Well, hello and welcome back to Science in the Age of COVID-19. Today we are super excited to welcome Michel Nussenzweig. He is a professor and Howard Hughes investigator at the Rockefeller in New York City. If you look on Michel's website, it says that his lab studies the molecular aspects of adaptive and innate immune responses with a focus on HIV. It is fairly comical how much this underrepresents the number of things that Michel's lab has worked on, both historically and currently. Michel has done a lot of work over the years that has pretty much laid the basis for the textbooks on the function of dendritic cells, the repertoire of peptides displayed by the major histocompatibility complex, and even with some big diversions into histones and their role in genomic stability. Of course, when COVID came onto the scene, Michel's lab immediately pivoted to that and has done so much on this virus that he will tell us about today. They've done antibodies, interferon response, genetics underlying that, diagnostics, you name it. I think he's going to cover 6 major papers today in his talk. And just one final note, when we were talking with Michel before the talk, he said that his lab has been working solely on COVID during this year. And I looked on Google Scholar and noted that they've still published 10 papers on HIV this year, despite only working on COVID. Michel, take it away.
Thank you. Thank you, and it's a real pleasure to be able to do this, and hopefully there'll be some questions and so on that come up as a result. So what I'm going to do is give a talk in several parts. I'm going to say something about background about immunology, which I think is important in trying to understand the human immune response to the coronavirus. I'll tell you something about why we were equipped to work rapidly on this problem, and then what we've learned in terms of human immunity and something about therapeutics as well. Okay, so this is what my lab has fundamentally been interested in for some time, which is a phenomenon that was discovered in the 60s by Herman Eisen and Greg Siskin, which is that the affinity of antibodies in circulation increases rapidly after immunization. And this is a log-fold change. It's a big change. People who have immunized animals have all seen this, and this is something that's true in vaccines and in humans as well. Now, we've learned a lot about this phenomenon over the last several decades. And one way to explain what happens is diagrammed here. So the immune system and the lymphocytes that make antibodies, the B lymphocytes, are clones of cells, each of which has a different receptor that's represented here by different colors. And when a virus or an antigen stimulates the immune system, what happens is that antigen selects for the B lymphocyte that has a receptor on its surface that can recognize the incoming pathogen or antigen. And that cell then begins to expand. And when it does something really very unique, which is instead of being a monomorphic expansion of cells, all of which have the same receptor and the same specificity, that cell turns on an enzyme called AID, discovered by Tasco Hanjo and Durandi, which makes mutations in the antibody genes in that cell. And so instead of having a group of cells that all look the same, you get a group of cells that are related to each other by small mutations in their antibody genes. And each of those receptors then has a different affinity for the pathogen. And then during the immune response, there is selection, and selection for cells that make high affinity antibodies. Now, this process happens in these beautiful microanatomic structures that live in lymph nodes and lymphatic tissues called germinal centers that my colleagues and I have been studying for some time. And these structures are compartmentalized. There are two compartments, a dark zone and a light zone, shown here. And the light zone here in pink is pink because the antigen is deposited in this zone on cells called follicular dendritic cells. So over the last several years, we've developed the model to understand or describe what happens here during this selection process. And I'm going to just go over it with you very, very quickly, but it's a relatively simple model. So there are two zones, and the dark zone is a zone where cells divide. And it's also the zone where cells turn on AID, the mutator, and make those mutations in the antibody genes. Once they stop dividing, this microanatomic compartment is highly dynamic, and the cells migrate, and they migrate massively from this dark zone where they've made the new mutations to the light zone where the antigen is deposited. And there they test their new receptors. So they test their receptors on the antigen. And if successful, if a cell has a sufficiently high affinity receptor, it will internalize the antigen, turn it into peptides, and present the peptides on MHC to specialized T cells that live in the light zone and that recognize the cell that has been successful because of the peptide MHC and send it back down to the dark zone. And there in the dark zone, the process will repeat. So a cell with the best affinity in the light zone will return to the dark zone, make additional mutations, clonal expansion, and move back up. And you can see that this relatively rapid process will lead to affinity maturation. So that's the working model. And there are two products of this reaction. There are plasma cells, which are the cells that are responsible for producing the antibodies that are found in the serum, and they are highly selected for affinity. But there's another product of this reaction, and the other product is the memory B cell, which has a long half-life, and unlike the plasma cell, is not so much selected for its high affinity, but it's selected in a way for diversity, for receptors that are mutated and have the capability of potentially recognizing not just the same antigen that comes in, but also variations of that antigen. And we'll get into that in the later part of the talk. All right, so why were we positioned to study coronavirus and rapidly understand something about the immune response to this virus? Well, we had been studying HIV for some time, and what we started out with was understanding that there was no vaccine and no cure, and despite that, there were some few individuals that were making very potent monoclonal antibodies or very potent serologic activity that could neutralize many strains of the virus. And we recognize that if whatever those people were doing could be reproduced, then we might be able to find out about the vaccine and why, in fact, it's been so difficult. And so we studied those people. And what we wanted to do was find out what antibodies they were making. And so what we did was to devise a method for finding the lymphocytes in the blood of those people that were producing the antibodies that we were interested in. And this is really a very simple method in the end. What it involves is the B lymphocytes have the antibody that they're making as a receptor on their surface. And so you can imagine that if you had the antigen available, which is in this case the HIV spike protein, that you could identify the cells that bound to the spike specifically, and then sort them as single cells and use molecular biology to reproduce the antibodies and test them for activity. And this sort of method has been adapted very widely, not just to study HIV, but also Zika, hepatitis, all kinds of different antigens and viruses and pathogens. In case of HIV, this has led to the finding that there are very, very potent antibodies that can be used clinically, and some of ours, in fact, have been licensed to Gilead for use in the clinic, and there are many ongoing clinical trials to test them. So when coronavirus came around, it was clear that the methods that we had developed could be useful in trying to understand the human immune response, in particular the human antibody response to this virus. And because most vaccines work because of antibodies, this was something that became very interesting to try to study. So what we did was, at the height of the pandemic here in New York City, we began recruiting people to Rockefeller to make donations of blood for us to study. We had 148 people that came to Rockefeller to do this. And what we focused on was the receptor binding domain of the spike protein of the coronavirus. And the reason that we focused on this, because it's this domain which interacts with ACE2, the cellular receptor that allows this virus to get into cells. And we felt that if we could find antibodies that would interfere with this interaction, then they might be neutralizing and teach us something about how to neutralize this virus. All right, so we initially started with just ELISA assays to look for what happened in people that were infected. And what we found was a relatively low level reactivity, but just about everybody had reactivity to the receptor binding domain. This wasn't something that we were particularly interested in primarily. We were really interested in neutralization. And so we worked with Paul Benash, who's also here at Rockefeller, who's also an HHMI investigator, to develop the neutralization screens for serum and antibodies. And what Paul's lab did was to insert the spike protein into an HIV pseudovirus, so that this virus now carries the SARS-CoV-2 spike. And we use that pseudovirus, which also expresses a nanoluciferase, to develop an assay which was linear over several logs for neutralization. And this assay we used to study plasma, and we compared it to actually authentic SARS-CoV-2 in collaboration with Charlie Rice, who's also here at Rockefeller. And you can see that the two assays are really very correlated, not just for plasmas, but also for monoclonal antibodies. So we feel very secure in using this assay on the samples that we obtained. What we found when we looked for neutralization in the cohort that we studied is that most people had relatively low levels of neutralizing activity. And you can see that here. These are the top 60 of 148. And this line represents 1 to 1,000 titers. And you can see that at least half of these people, the top 60, fall below that. And then the rest, of course, are even lower. So generally, there is neutralizing activity, but it's not really very high. Now, the neutralizing activity in serum was dependent on the ability of the antibody to cross-link the virus. And you can see that here in several different samples where we compare the IgG and the FAB digested from the IgG in serum. And in all cases, there's at least about a 10-fold drop in activity when you use the FAB, which means that you really need to cross-link something in order to get optimal activity. And this result has later been confirmed by many labs, including Peter Walters, who developed some nanobodies, which when he makes them divalent, become much more potent. So summarize what we learned from serology. So there are low levels of antibodies to RBD in most infected individuals. And there are low levels of neutralizing antibodies in most infected individuals. And cross-linking was required for optimal neutralizing activity. We also did some clinical correlates that were interesting. What we learned from them is that neutralizing activity correlates with the duration of symptoms, symptom severity, and age. Those are all codependent variables. So people who are older are sicker longer, so duration of symptoms. They generally have more severe disease. Again, all of those things are codependent. Men have a higher neutralizing activity than women. Again, this is just the same kind of thing. Men are sicker longer, tend to be sicker longer. And hospitalized people also have higher neutralizing titers. And all of that goes together to say that people who are exposed to the virus for longer periods of time have higher serologic activity. Now, having this cohort and the samples that they donated allowed us to select individuals, basically all the ones with little red stars here, for antibody cloning, to learn something about what kinds of antibodies that we're making that were responsible for the neutralizing activity. We did this just the way that I described, using the samples that they donated, staining the lymphocytes in the blood with the viral protein, taking cells that stained, single cell sorting, and then just traditional molecular biology to reproduce the antibodies. This is what we found. So here is a control being stained with the RBD in two different colors. So the cells that should be positive would be here, and you can see that the controls are negative, but every single person that we examined, whether or not they had high neutralizing activity or not in serum, had memory B cells that stained for the RBD. When we pulled out those cells and did the cloning, we got what's represented here in these pie charts. Each pie chart is one individual. The number in the middle is the number of antibodies that we cloned out. The pie slices represent the size of the expanded clone. So remember, during the immune response, there's clonal expansion, and these are very closely related members of the same clone. The slices that are colored indicate antibodies that are nearly identical in different individuals. So you can see that different individuals are solving this problem in related ways. Now, in doing this, we found many antibodies that were very, very potent, including these three that are tested here against the pseudovirus and authentic SARS coronavirus. And you can see that the IC50, so the ability to neutralize 50%, is in the nanogram range. So that's a very, very potent antibody. Now, we wanted to learn something about the mechanism of neutralization. And so we collaborated with Pamela Bjorkman at Caltech on this, and her postdoctoral fellow, who's an HHMI fellow, on learning something about the structures. What we learned is shown in the next several slides. So the first slide illustrates what we call a class 1 antibody. So this in gray represents the receptor binding domain, and the antibody is shown here in green. This class of antibodies binds to the receptor binding domain when it's in the up configuration. And what I should have said on the previous slide is that it's only in the up configuration that the RBD can bind to ACE2 to access the cell. So this thing binds the up configuration. It binds to the same ridge on the receptor binding domain as ACE2. So it just blocks ACE2 by directly interfering with that interaction. Now, this class of antibodies includes some which repeat in many individuals. These antibodies that are repeating in many individuals use this loop, the CDR2 loop, in a major way to interact with the RBD. And that part of the antibody is encoded in the germline. It's not made by VDJ recombination, which explains why many individuals will use this early on in the infection to protect themselves against the virus. A second class of antibodies also binds to the ACE2 binding ridge shown here, but the spike is a trimer. And in fact, this class of antibodies can reach over to the next door trimer. It does that by projecting a loop with some hydrophobic residues into a hydrophobic pocket in the next door receptor binding domain. And the result of that is really something interesting, which is that it locks down the 3 RBDs in the trimer in a position where they cannot move up and access ACE2. So this blocks ACE2 in 2 different ways. It blocks it by interfering directly by binding to the ridge that ACE2 binds to, but it also prevents the RBD from moving up and accessing ACE2. So 2 mechanisms of interference with infection. There's a third category shown here, illustrated here, and this category does not directly interfere with ACE2 binding, but in fact binds below the ACE2 interaction ridge and interferes indirectly with ACE2 binding. All in all, there are 4 classes that Christopher has defined 4 classes of antibodies that bind to and can neutralize the virus by binding to the receptor binding domain. Now, if there are classes that are potentially complementary, like class 1 or 2 and class 3, then perhaps they can be used together in order to prevent the emergence of escape mutations. Before I go into that, actually, I wanted to bring you up to date on what happened after 6 months. So that was what I just discussed, was what we found after 1 month. And so after 6 months, we brought people back because we were very interested to understand whether or not their responses were lasting. Clearly, they had neutralizing activity. Clearly, they were making good antibodies. What happened later on? All right, so this is a paper that is up on bioarchives and is currently being reviewed that I'm going to talk about next. Each one of these dots is an individual, and this is their neutralizing activity in blue after 1 month, and then after an average of 6 months in red. And you can see that it's sort of a parallel line. On average, people are dropping their neutralizing activity by about a factor of 5 after 6 months. But in just about everybody we tested, we can still see some neutralizing activity. So that's the product of the plasma cells. What about the memory cells? And the memory cells are what's going to be important in responding to a subsequent challenge. There are two kinds of memory cells. There are T cells and B cells. In this other manuscript, what we do is describe what happens to the T cell responses. And you can see that very clearly, I think, here in this multidimensional flow cytometry plot. So here are controls that have been stimulated with a series of peptides from the SARS coronavirus. And if you look just in this area here, pointed out by the red arrow, you can see that there's a big difference in the recovered individuals. So they have memory responses to those peptides, and those responses are waning, but still present after 6 months. And you can see that in the graph below as well. So the controls are in black, and you can see that here there's a substantial response, which also decreases, similar to what you see in the neutralizing activity in plasma. But memory cells are there. The B lymphocyte memory, we assayed using the flow cytometry trick that I talked about earlier, which is to label the receptor binding domain directly. And you can see that here for the initial 6 individuals and aggregate for 20 individuals here. And what's surprising is not only were the B memory cells retained, but in fact, they seem to be, in fact, increased. So these guys are really quite stable, and they're going to be producing the antibodies upon rechallenge. If we look at each individual antibody, so this is after cloning, you can see that some of the clones are retained, and that's these colors, although generally the clones are decreasing in size. There are also clones in gray that disappear at the second time point, and new clones in gray that appear at the second time point. And overall, the amount of clonality decreases in a quite significant way. Now, these antibodies are, in fact, one of the surprises was that these antibodies are evolving. So initially, the antibodies that we found had very low levels of somatic mutations. Remember, the somatic mutations are occurring in the germinal center. And it was thought that there were defective germinal centers, which is why we were seeing so few mutations in all these individuals. But in fact, after 6 months, you can see that in every single person, highly statistically significant increases in the numbers of somatic hypermutations. So there's an evolution in the clones, and there's an evolution in terms of somatic mutation. And there's an evolution in terms of the potency of the antibodies shown here. So a highly statistically significant increase in the potency of the antibodies that are retained after 6 months. And you can further see how that evolution is playing out here in these two sets of antibodies. So the green antibody is the antibody that we obtained after a month or so, and the red one is something we obtained from the same clone, but after 6 months. These 50 or so mutants represent circulating mutants and escape mutants that the RBD puts up. And you can see that the red antibody is, first of all, far more potent. And also, the red antibody is resistant to mutations that completely wipe out the activity of the 1-month antibody. This one, 144 in green, which is the 1-month antibody, is a really excellent antibody, which is in fact resistant to most mutations, but not all. And you can see that the 6-month antibodies actually acquire activity which makes them resistant to RBD mutations. So evolution in terms of somatic mutation, evolution in terms of clones, and evolution in terms of potency and breadth. So something is going on for a long time. And you can see here from this analysis of the T cell compartment, which includes not just the antigen-specific T cells, but overall, that there are continuing abnormalities in these individuals. So you can see, if you look at the arrows, this one is easier to focus on here, you can see that after 1 month, it's really very different in these individuals. And after 6 months, it remains different from control. And these differences are highly statistically significant. They mainly relate to the memory compartment in these individuals. And we think that what's going on is that there is some residual antigen, that the residual antigen seen here in immunofluorescence in people that are between 1 and 4 or 5 months out from their infection is in the gut in cells that express a lot of ACE2. We even think that there is some residual virus and not just debris shown here in an EM. So for the long term, memory B cells persist after 6 months. The antibody responses are evolving, become broader and more potent. CD4 T cell responses also persist after 6 months, and there are some abnormalities that persist in the T and B cell compartments. So there is something, antigen, we believe, in the gut for several weeks and months after infection, which may account for this persistent evolution. So in summary, individuals retain neutralizing activity for 6 months, but it's 5-fold decreased. The memory cells persist. Antibodies expressed by memory B cells are improved. And overall conclusion would be that immunity appears to be lasting after this coronavirus infection. And that's something that people debated quite a bit, but in this case, it's very likely that if an individual was infected and did well, they're going to do well when they are re-exposed again, and really for quite some time. All right, so in doing all of this, we discovered a series of antibodies that are really very, very potent and complementary. What's the potential use for these things? Well, first of all, they could be used for protection. And I'll just show a couple of experiments that support that idea. And there will be individuals that cannot respond to the vaccine for whatever reason. It's people, for example, that are immunocompromised, the very elderly, and there's just a whole huge group of these people. 5%, even if the vaccine is 95% effective after a year, which is extremely unlikely, 5% of 300 million people is still a lot of people. In addition, the antibodies can be used for therapy, and this has gotten a lot of play in the news. Because our outgoing president received them. And again, this can be a very large group of people that would benefit from this, including those people that fail to respond to vaccines, others that refuse to take vaccines, and others that just don't make a good enough response to the vaccine. So how do we know that these things will be effective? Well, we are beginning to learn that from clinical trials, but also from experimental animals, and probably the best experimental animal for this, or the most difficult one, is the hamster. And here are 2 experiments where we delivered monoclonal antibody combination to hamsters either before infection or after infection. This is a very rapid experiment because the peak of infection here is day 3. And after that, the animals begin to lose weight and do poorly. So this experiment has a pretty good dynamic range. It's a measurement of the plaque-forming units, so real virus in the lungs. And you can see that in a sham animal, it's 10 to the 6th, about 10 to the 6th. And then in the prevention experiment, you can see that 20 milligrams per kilogram, 6 milligrams per kilogram, or even a dose as low as 2 milligrams per kilogram is highly effective in prevention. In therapy, we have very similar results with 40, 12, and 4 milligrams per kilogram of a combination of 2 antibodies. So these antibodies can both prevent and treat infection, and they can treat infection if they are administered early. This is in macaques. It's a very similar result with even relatively low doses of antibodies being able to prevent infection. And this is therapy experiment in macaques, where the readout is pneumonitis, and that's scored by 2 independent pathologists in a blinded fashion. And again, there's a highly statistically significant effect of the antibodies. So for clinical applications, what's required is safety, potency, a long half-life, resistance to escape mutations, and something practical in terms of route of administration, which is not intravenous, but sub-Q or IM. And we have been developing a combination of 2 antibodies together with Bristol Myers Squibb. So in terms of potency, these are the ones that are being developed, most prominent ones. And the Regeneron and Lilly are the ones that you've heard most about. And here are their potencies, their published potencies, and confirmed these. And these are the 2 Rockefeller ones, Rockefeller Bristol Myers Squibb's HHMI, actually, antibodies. They're a bit more potent than the rest. And they also carry mutations which triple their half-life. So they're very long-lived antibodies as opposed to the Regeneron antibodies. And where are these antibodies in terms of their clinical use? The first in human safety studies will be performed here at Rockefeller in January. They will enter the phase 2-3 NIH-sponsored trials in March and April. We're also going to test them in people that have immunodeficiencies that can't respond to the vaccine, both for protection and therapy. And we hope that we'll be able to get them to people in May or June. So what I've talked about is an effort that we started in April of this year, which is 3 or 4 months later than Regeneron or Lilly began their efforts. But we've learned a lot in terms, not just in terms of producing antibodies for the clinic, but also in terms of the biology of this disease. We learned about how humans react in terms of their antibody responses, and something about why there are similarities between different individuals, and also generally how the antibodies neutralize the virus. And we've obtained some reagents which we think can be of significant clinical benefit in what's bound to be an ongoing problem in the future. So this work is the work of many people, but led in my lab by several individuals, Davide Robayani, initially, Gael Breton, Shlomo Finkin, Vinci, a whole series of people in the laboratory have worked on this. We have a clinical group at Rockefeller headed by Marina Kasky, and Christian Gabler has been the primary moving force there in bringing this to the clinic. We've worked very closely for many years with Pamela Bjorkman, first on HIV, then on Zika, and now on the coronavirus. The BNASH lab and the Rice lab, virology labs at Rockefeller, have been instrumental in developing the assays that we use to assess the antibodies. And a whole series of other laboratories all over the country have participated in this. It turns out to be, quite a huge effort, actually. So I'm happy to take questions if there are questions.
Thank you. Thank you, Michel. That was a great talk. And let's get right into the questions. Our first question is from Kaylin Shireen, and I'm going to read this out for you. Are there data to show whether one or more subtypes of IgG is more involved in the response than other IgG subtypes?
Yeah, generally these are IgG1s, and they're the dominant IgG in human serum. There are IgA responses as well, and those are the responses that are most prominent in the mucosa. And they can be particularly effective because IgAs exist as dimers, so a dimer of a bivalent antibody. And in the case that you make a dimer of a bivalent antibody, you increase the valency. And by doing so, you also increase the potency. So vaccines, for example, that elicit mucosal responses, IgA responses in particular, may be very effective because these multimers really increase the valency.
Thank you. While we are still on Ig immunoglobulins, here's the next question. This is in response to your observation that IgA responses are higher in individuals with GI symptoms. Mm-hmm. Is there a reason that you see this?
Yeah, so the mucosa, the GI tract is lined with immune cells, and those immune cells tend to produce mucosal antibodies, IgA. So people that have GI symptoms clearly have antigen in the GI tract, and that elicits this kind of response.
Michel, were you guys measuring IgM at all in your studies?
Yeah, we did look at IgM responses. IgM responses tend to wane the quickest, which is expected. And you can see when you do the antibody cloning, you can see related clones of B cells that express essentially the same specificity that is an IgM, an IgG, or as an IgA.
All right, thank you. We have an audience question now from Tim. Tim, please unmute yourself and ask your question.
Oh boy.
Tim, you sound like Alvin and the Chipmunks. Maybe remute and unmute and try again, or else type your question because we can't understand you.
Change phones and then change back.
There we go.
You're good.
Michel, I wanted to ask about this frequently repeated comment by television physician commentators that if you have been immunized, that you are in no guarantee of not having a large viral load and therefore infecting people, and you must continue, to wear a mask indefinitely, apparently. That seems to me to be nonsensical. If you're not diseased, can you have a high viral load and infect someone else?
Well, okay, so what this is about is the way that the vaccine was actually tested. So the 95% comes from, thousands of people that were given the vaccine, and then they were asked, if you have symptoms, come and see us and we'll test you. So only people that were symptomatic were tested. So that's where the 95% comes from. Okay, so if you were infected but asymptomatic, you would not be tested. And it is entirely possible, given what we know from the macaque studies, that people would be infected in the nasopharynx, but not come down with pulmonary disease, which is what would make them symptomatic, okay? Or, start coughing or what have you. So in the monkey studies, the vaccines gave protection as measured by decreases in virus, but it was not sterilizing. Okay, so the vaccine was not sterilizing in macaques. Antibodies, when you give them, are typically not sterilizing in macaques, most antibodies, although some will be. So yes, it's entirely possible that you will be infected, that you will have virus, but you will be asymptomatic after receiving the vaccine. So you will be protected from disease. That's what this vaccine does. But you will not be entirely protected from infection.
Are you good, Tim?
Okay, thanks.
Thank you, Tim. Thank you, Michelle. A little follow-up question. So you showed that immunity lasts after infection. If so, how would you explain the cases of reinfection? I mean, are they escape mutants or?
Well, no, I would say that first of all, those are extremely exceptional, right? Just think about it. How many millions of people have been infected and re-exposed? And essentially, you can count the number of well-documented cases. So a very, very tiny fraction, and there's probably something about those individuals which is different. But most people will not.
Mm-hmm. Thank you.
Hey, all right, Michelle, I'll go now. I've got a ton of questions. That was fascinating talk. So yeah, the various antibody cocktails have been, all over the news. They're very subject of many memes at this point. But frankly, a lot of us are pretty disappointed by the efficacy. Both Regeneron and Lilly cocktails. Small N so far, but they haven't made a difference in hospitalizations, ER visits, and so
they will make so they make a difference. Where they make a difference, where antibodies make a difference, is in the early phases of this disease. So if you are hospitalized, by the time you're hospitalized, it's no longer about the virus. Mm-hmm. It's about the damage that the virus has caused. So if you are on oxygen and you are 10 days or 2 weeks away from your initial infection, the virus has done its damage, and there's very little the antibody can do. So where the antibody will have an effect and has an effect is early on. So like Trump, he got his diagnosis probably on Tuesday before the debate. And he got his antibody therapy on Thursday or Friday. And that is exactly when it will make a difference. It will essentially prevent the worst disease, and if given at the right time, will prevent hospitalizations. So it's a question of timing. It's not going to help you when you're past, a certain point.
Yeah, I mean, are you aware that this is that how it's being used?
That is how people are thinking about using it, yes. And that is how the emergency use authorization was granted, is basically keeping people out of the hospital. Yeah. So, and that's a critical thing in terms of, the hospital usage. is keeping people out. And that's how it will be used. That is, so there will be really two uses for this. Protection in people who are unable to make a response to the vaccine. And then those people who, for whatever reason, get infected and diagnosed fairly early. So you can imagine that, there will be a home test very soon at CVS. And people who have a cold or whatever, feeling like they have a cough, they'll take the test. If they're positive, they'll go to their doctor's office or wherever it is and get a shot of antibody. And that's going to be the use of this thing.
Excellent. So we'll bounce over to another audience question. Caitlin again says, CDC has issued guidance that it can take a few weeks for the body to build immunity after vaccination. What are the data on which this timeline is based, and is this an accurate timeline?
Yeah, so the data is based on the separation curve from the Pfizer vaccine. So they have a control group and they have a vaccinated group. And what you see is that the two groups begin to separate 2 weeks after the first injection, and that there is 50% protection between the first injection and the second injection, which happens at 4 weeks. And that it goes up from there. So there is some protection after the first injection, beginning around 2 weeks, which is the time that it takes to make an adaptive immune response. And then it increases thereafter.
Excellent, thank you. Okay, so Sarada's got one next.
Yes. So, Michel, this is regarding the residual antigens that you observe. So you showed that you see residual antigens in the gut, but we know that the coronavirus affects a range of tissues. So are there any other tissues that you observed residual antigens?
No, this has been done in postmortems, the other organs, but we were dealing with people that were alive. And so it's not easy to get donations from other organs. The gut is a particularly easy place.
Understandable. So, and you mentioned that it's an intact virus that is possibly, that accounts for persistent evolution of the antibody response. So do you think you would need an intact virus to evoke this?
No. So, just debris, just a little bit of antigen goes a long way. And I remember that picture that I showed of the germinal center with the pink stuff in the light zone. Those aggregates, those deposits remain on the surface of those follicular dendritic cells for long periods of time because those cells are not endocytically active. So they just hold on to this stuff for as long as, it takes for the B cells to pull it off. Which can be quite a long time.
Thank you.
Michel, I have a related question. So, very compelling evidence that there is some pool of antigen hanging around, and maybe live virus. I was struck by the differences in the long-term responses between B cells and T cells. B cells are continuing to evolve their antibody repertoire to tamp down antigenic escape, whereas the T cell response seemed kind of frozen at that 1 month timeline. Do you have any insight as to why that is?
No, I don't. And in fact, the memory T cells drop off, whereas the memory B cells seem to, in fact, increase. The way those things are produced is really very different. So the germinal center, if it's ongoing, which it must be because of the accumulation of mutations, it's going to be throwing off memory all the time. The T cell isn't like that. It's dependent on antigen presentation by dendritic cells in order for its clonal expansion. And it doesn't have, and the dendritic cell is an endocytic cell that will destroy the antigen as opposed to the FDC, which will just hold on to it. So destruction of antigen in one case versus retention of antigen in the other may explain some of this.
Yep, that sounds reasonable. Let's see. Yeah, we have an audience question. Yeah, it's asking about the logistics of distributing the monoclonal antibody there.
Yeah.
Do you think we'll be able to pull it off?
Yeah, well, that's logistically a lot easier than, minus 70 or 80 or 90 or whatever it is, because these things are stable at 4 degrees, and so it's pretty standard. And as I said there, in terms of practicality, the way they're currently being used is intravenously, which is not very practical, but because it requires an infusion chair in a specialized location. And how do you bring somebody that's infected into that location where typically you're treating cancer patients or something else like that, which are very, very susceptible to the coronavirus. So there are all kinds of logistical issues with intravenous administration. But some of these, the ones that Bristol Myers, the HHMI, Rockefeller, Bristol Myers antibodies, they are subcutaneous delivery. So that's a lot easier.
Yeah, and one of our speakers was the lab that made the nanobodies that they're thinking of delivering as an aerosolized nasal spray.
Yeah.
And there's it's not an antibody, but it's a lipopeptide mimic of the virus that just seems it confuses it and thwarts the virions. That seems effective as a nasal spray. I think that was in ferrets. But I think that's way far behind in the clinical pipeline.
Yeah, those things, they're going to be good solutions to this thing. Better and better and better solutions.
Yeah. Strati, did you have another one? I've got some more.
Why don't you go ahead? I will follow up.
Okay. Michel, so we've got this persistent B cell activation in the germinal centers. Somatic hypermutation is picking up. You can probably tell where I'm going. So, autoimmunity has been shown in a lot of people with long COVID, people transitioning into arthritis, et cetera. I mean, of course, it'll be a long time before we can really nail this down. Do you have any insights? Do you think that there are specific sequences in the virus that are the mimetics, like we think for Epstein-Barr, or what do you think?
I'm not, first of all, I'm not convinced that long COVID has anything to do with autoantibodies.
Really?
I just don't, I'm just not convinced.
Okay.
And there are many potential explanations for the long COVID. A lot of viruses are associated with prolonged malaise and just really not feeling particularly well. One of the explanations for that I've been thinking about is that if you look at some of the slides that I showed, you can see a persistence of T cell abnormalities many months after the coronavirus infection. And that's in just about everybody. And it may be, and human beings have different sensitivities to different things. So, some people are very sensitive to salt. Some people are very sensitive to sweet. Other people are very sensitive to noise. And it may simply be that some individuals are more sensitive to immune abnormalities than others. So for me, that's as good an explanation as any current explanation for the long corona thing.
Yeah. To be clear, I was not suggesting that autoantibodies,
Yeah.
completely explained long COVID, just that they could be, one of 20,
Yeah.
20
Yeah.
things.
Yeah, I mean, it just isn't clear yet what that's all about. And I don't think, the germinal centers are always ongoing in everybody because we're always being challenged with something. So I don't think that they're particularly generating anything unusual in this case.
Okay, I hope you're right.
Me too. Me too.
Yeah, and Caitlin, who's a immunological expert, just writes that with all the long GI symptoms that some people see, she's just noting that there is a lot of overlap with celiac style IgA and IgG persistent response. So yeah, I hope you're right. But as a person with autoimmune conditions, I have to say that I'm very worried.
Yeah, I wouldn't be. I wouldn't be too worried about that, but do get yourself vaccinated.
Oh God, yes. Okay, Michel, this has been amazing, and thanks so much for your time and all the work that your lab has done. So Matt, can you show the slide for next week, please? Yeah. Oh, looks a little.
Yes.
Okay.
Yeah.
I'll bring out the slide.
Okay, very good. I'm going to sign off. Bye-bye for now.
Okay, Michel.
Thank you.
Bye.
Take care.
Thank you, audience. Thanks for joining us. This was the last talk of this year. So we will all see you next year with many interesting talks lined up. The first talk will be from Dr. Scott Hensley from Penn Institute for Immunology. So happy holidays. Stay safe. See you all next year.